Hello, everyone. My name is Gladys Alexandre. I'm a professor at the University of Tennessee, Knoxville, and I'm really happy today to share some work my lab and many other groups have done on characterizing bacterial chemotaxis in beneficial plant microbe association. This work is rooted in a challenge that the world is facing, and that challenge is we will have to feed about 10 billion people by 2050. That increased demand for food would require about 50 to 60% increases in agricultural production, according to a National Academy of Sciences report. And most of this increase in food production would have to be done in a sustainable manner or using sustainable agricultural practices. So plants have diverse microbiome associated with their tissue, whether they are below ground or above ground. And this tissue represents the phallosphere, or the surface of the plant, the leaf endosphere, which is the intracellular compartment of the above ground tissues, the rhizosphere, or the zone around the roots, and the root endosphere, which are the tissues within the root. An observation that is made about these different habitats is that they differ structurally and also in terms of their chemical environmental conditions. So, for example, the environment around the leaf surface is going to be very different from the environment around the root in the soil. And these diverse conditions provide habitats for bacteria and fungi. And an observation that have been done in many studies is that the diversity of bacteria and fungi that you find in the bulk soil is far greater than the diversity of bacteria or fungi that you find in other plant tissues, such as the phytosphere or the rhizosphere. And that has led to the hypothesis that plants select for their own microbiomes, and that they select or particular microbiome have property to specifically colonize certain plant tissues. So the rhizosphere, or this region of around the roots, is an heterogeneous environment in terms of structure. For example, here on this image, you can see that you can have very thick and very thin roots, and also in terms of chemicals. Many studies have shown that plant roots can secrete a diversity of compounds, such as sugars, amino acids, organic acids, phenolics, as well as hormones that can be recognized by microorganisms that live in the rhizosphere. Some of these compounds can be utilized by microorganisms that live in the rhizosphere. And these chemicals modulate plant microbe association as well as microbe associations. So the rhizosphere is heterogeneous, not only physically, spatially, but also chemically. So manipulating rhizosphere microbiomes is one of the potential solutions to meet sustainable agricultural goals for feeding a growing population on the planet. And the reason for this strategy is because the rhizosphere microbiome is critical to sustain plant health and productivity. And that has been shown in many studies, and the mechanisms involved are very diverse. For example, some members of rhizosphere microbiome can produce compounds that will protect against phytopathogens. Others can produce or modulate plant growth hormone signaling or production, and others can also improve nutrient and water uptake by the roots. Many of these mechanisms are produced by multiple organisms, or sometimes by the same microorganism. So plant growth promoting rhizobacteria, or PGPR for short, are microorganisms that have been proposed to be used for sustainable agriculture practice. And the reason for this is that these rhizobacteria, bacteria that live in the rhizosphere, can colonize plant roots and promote crop productivity. And they do so by a variety of mechanisms, some of which are listed here on the slide. So it could be nitrogen fixation, hormone production, siderophore production, phosphate solubilization, increased nutrient availability, or by promoting induced systemic resistance in the plant. And this mechanism can be used in combination by some organism, or only a few of them could be used by others. But together, these plant growth promoting rhizobacteria benefit plant growth and in turn improve soil fertility. As a result, a lot of these PGPR have been developed and are now commercialized as biofertilizer or bio-inoculant. And they do work quite well. Here I'm showing you two examples. On the top is a field of corn or maize. On the left-hand side, they've been inoculated by a PGPR, and you can see that the plants are greener and taller and look healthier. On the bottom image, these are pea plants that were inoculated as well, and you can see obviously that the inoculated plants look healthier than the non-inoculated one. So in order to produce their beneficial effect on plant, the PGPR or any bacteria that is present in the soil must colonize the root surface. And in order to colonize the root surface, the bacteria that are present in the soil must first detect that a plant is present. In other words, sense that the plant is there, and then actively move toward the plant root surface. And the active movement in the direction of the root surface is called chemotaxis. My lab has been using two different plant growth promoting rhizobacteria to study chemotaxis and the role of chemotaxis in association with the plant. And so one of them is called Azospirillum brasiliense, so diazotroph, nitrogen-fixing bacteria, and PGPR. And the other one is Rhizobium leguminosarum biovarviciae, so legume symbiont. Both of them have been used around the world in several biofertilizer or bio-inoculants and have been quite successful. So they are pretty good model system for at least my research goal, but many others as well. Okay, chemotaxis in bacteria consists of biasing a random walk. And that's because bacteria are too small to sense a gradient along their cell body. And instead, they have to make temporal comparisons, meaning they compare the conditions now to what they were a few seconds before. And so in the absence of a gradient, the bacteria have what we call a random walk, where they alternate period of smooth swimming with random change in swimming direction, and their net movement is absolutely nowhere, and they stay in the same area. In a gradient of an attractant, here to the right, the cell will tend to swim longer in the direction of the attractant and suppress change in swimming direction, and their net movement will be up the gradient of attractant. Conversely, in presence of a repellent, here also to the right, the cell will change direction very often close to the repellent, but swim straight away from it longer, and their net movement will be down the gradient of the gradient of this repellent. At the molecular level, this chemotaxis is controlled by a signal transduction pathway that has been best characterized in Escherichia coli. And this pathway involves chemoreceptor, transmembrane chemoreceptor that sense attractant and repellent. These receptors are associated with cytoplasmic protein, which ultimately will control the direction of rotation of the flagellum motor. The major protein controlling that entire signal transduction pathway is the chemoreceptor protein A, or Ki-A. And chemotaxis systems have been best characterized in E. coli and other model systems, but they also have been studied in metagenome. And what we have found using these studies, chemotaxis function is enriched in bacterial metagenome found in the soil. So metagenome are the total genome or total DNA you could extract and sequence for a particular sample. And if we take soil metagenomes database and we look for Ki-A, which are essential for chemotaxis, and we count how many Ki-A compared to how many genome are representative in this data set, we can see that over 99% of all genome represented in soil metagenomes over 99% of these have more than 2 Ka per genome, meaning they potentially have more than 2 systems like this one in each genome. Compare this with the human gut, where only 10% of the genome represented in that metagenome datasets have only 2 Ka per genome, so it's less enriched. And in the ocean, most bacteria will not have chemotaxis system. Only 2% have more than 2 Ka per genome. So these observations are consistent with chemotaxis being a function that is important in the soil environment. How do we study chemotaxis? There have been several assays that I will present really briefly. One of the most utilized assays are spatial gradient assay. The first one is the swim plate assay. In this assay, cells are inoculated at the center of a soft agar plate that contain compound that these cells can metabolize. By metabolizing this compound, the cells deplete the local environment and therefore create a gradient. And if they are motile and chemotactic, they will be able to move up this gradient and form these concentric rings that you can see here after a period of incubation that's about usually at least several hours. A variation of this assay is the capillary assay. In this assay, you place the attractant or the repellent in a capillary tube that you then place in a pool of motile cells. And then you measure, after either minutes or several hours, the accumulation of cell around the capillary tube, for example, using microscopy and image analysis. Or an alternative way is to wait a little longer and to count how many cells have moved up into the capillary and counting them over time. So this can be a little longer. A much more direct way to measure chemotaxis corresponds to temporal gradient assay. In these assays, you directly use computerized motion analysis to track free-swimming cells and their response to addition of an attractant of a repellent. So here is an example where you have tracks of free-swimming motile bacteria with the arrow pointing to examples of change in swimming direction. If you add a stimulus, for example, here an attractant, and you continue tracking the cells, you will see that now these cells that had a random walk now suppress change in swimming direction and swim smoothly. After a while, they will return to this pre-stimulus response, and it usually takes a few seconds. But the time it takes to return to this pre-stimulus response indicates the strength of the chemotaxis response. So it can be a very quantitative assay. So obviously, this assay works well for model system and in the lab, but it's not necessarily the easiest to characterize plant microbe associations. And in fact, what has been done classically to characterize the role of bacterial chemotaxis in plant microbe association is to use culture of bacteria that you want to test, maybe even mutants, and you inoculate them to plants. And then you wait several days or few hours, and then you collect root tissue and you count how many cells were able to colonize. Obviously, that takes can take several days. The problem with these assays is, as I indicated earlier, chemotaxis is a really quick response. It takes a few seconds. So by just inoculating and counting and taking few hours, a lot of things have happened between the chemotaxis response and what you're measuring after these several hours. So we were interested in developing assay that would allow us to track chemotaxis response in real time. And my laboratory was especially interested in looking at how cells behave in different regions of the root. So the root hair, where there's a lot of absorption of nutrient, the root elongation zone, as well as the root cap or root tip. And what we did is we developed a slide-in microscope chamber, which is a 3D printed a chamber in which you can plant little seedlings. And the seedlings will be incubated in a gel that supports plant growth. And you can also inoculate bacteria to this. And you have little grids that allow you to determine the location of your observation, but also to see through and come back several times to see the same area of the root. The advantage of this plant growth chamber is you can image the same region of the root over time without having to destroy your experiment every time to count cells on the root. And so we used this to look at cells. And here I'm showing you one of the earlier movie that we had. It's a real-time movie. This is a root hair. And you can see that cells are swimming around the root hair. And you can see that here they form a band where they seem to be stuck in this region, and some other are swimming more freely around the root hair. So that was an earlier observation, but we also did this by looking at cells that were fluorescently labeled, here green. And as you can see here, this is a time lapse. You can see that, again, cells form some kind of a dense band away from the root hair, and that cells in the middle are able to move freely. So we use that chamber and these assays really to characterize the chemotaxis response of Azospirillum brasiliense to wheat root hair. And as we could see different pattern of colonization for the wild type. So here in red are plant tissues, they are autofluorescent, and you can see that the green bacteria really colonize the surface of the root hair. They also are able to colonize the surface of the root elongation zone. The root is here, but you cannot observe any bacteria at the root tip or the root cap. We know this response depends on chemotaxis, because if we use a chemotaxis mutant, so this mutant cannot detect signal or bias its motility, but it is fully motile, you can see that the cells are randomly distributed regardless of the region of the root. We also analyze a receptor or chemoreceptor mutant lacking the TLP1 chemoreceptor. So it's lacking one of 51 chemoreceptor encoded in the genome. And we can see that the pattern of colonization is very different from that of the wild type, suggesting that this chemoreceptor is important for recognizing different zones on the roots. In addition, using this small chamber and this observation over time, we were able to show that the bacterial accumulation in different root zones precedes colonization. And what I mean by that is 24 hours post-inoculation, here we can see that cells that are fluorescently labeled in green again, they accumulate as large band at some distance from the root, but they do not yet colonize. For example, here the band is about 150 micrometer. When you wait another 24 hours, now the cells the band is no longer observed, and the cells are found on the root hair. And you can see now also they are found on the root elongation zone. If you look at the root tip, there is absolutely no colonization, and we actually can quantify this pattern of colonization and see there's very little colonization of any of this area at 24 hours, and a lot more of the root hair and root elongation zone at 48 hours. So we know that depends on chemotaxis, because if you use this non-chemotactic mutant that is fully motile but cannot detect signal in the environment, we can wait over time, we never see any colonization of any of this area. So if this behavior that we see here on the plant depends on chemotaxis, then we should be able to see a similar pattern with an assay that's even shorter term assay. And for that, we just adapted an assay that resembled the capillary assay, but instead of using the capillary, use an entire root, and we put it in a pool of motile bacteria, and then we measure accumulation around the roots of plants within 5 minutes of placing the root into that pool. And what we can see, if we use, so this is the root here or here, and this is the root hair zone, and we place in a pool of motile cells, we can see that there's a band forming over time. This is a time-lapse experiment, and you can see that that band moves toward the root hair. On the root tip, the wild type behave differently. We can see there's a band forming at some distance from the root tip, and it always stays away from the root tip, and the band even becomes thicker. We know it's dependent on chemotaxis, because if we do the exact same experiment with a non-chemotactic mutant, we do not see any formation of any band. Therefore, this short-term chemotaxis assay seems to indicate that indeed chemotaxis is important for recognizing different regions of the roots. And so our hypothesis that was the cells could sense root exudates. So we went back to this chemoreceptor mutant because we know this chemoreceptor sense organic acids. And what we could see is that if you miss one of these chemoreceptor out of 51, now the pattern of colonization is very different from the wild type, which is shown here in quantitative assay. And in fact, the mutant seems to be colonizing now the root tip a lot more than even the wild type that did not colonize it at all. So we also use the short-term assay and confirm this. The wild type formed this band away from the root tip or closer to the root hair. The chemoreceptor mutant didn't seem to sense the root tip at all and form a very, very faint band that did not move over time in the root hair zone. So obviously, sensing organic acid by TLP1 is important for this behavior. So this chemotaxis defect suggests distinct chemical condition in different regions of the roots. We were particularly puzzled by the response to the root tip because the root tip or the root cap are known to produce reactive oxygen species. And this has been demonstrated in different studies. And we know that azospirillum, in fact, increased that response. Inoculation with azospirillum increased that response. So we had to adapt yet another assay to try to measure chemotaxis response to a compound that that is similar to a reactive oxygen species, and we selected hydrogen peroxide. And what we did is we took a disc saturated with hydrogen peroxide and put it on top of a soft agar plate in which wild-type cells were inoculated. And we saw within 20 minutes that these wild-type cells moved away from the disc. If we place the disc that's saturated with buffer on the same plate, the cells did not respond at all to this. Mutant lacking TLP1 did not respond. And if we express a parental receptor into the mutant, we can restore the response we see in the wild type. And we know that was very specific to hydrogen peroxide because if we use cumin hydroperoxide, which is an organic peroxide, generating stress, we do not see this response at all from the wild type or the chemoreceptor mutant. So together, these data that we have shown you show that in the root hair, azospirillum at least, detects some attractant, and at the root tip, it detects some repellent, perhaps ROS, and that these different recognition sets colonization preference. So the fact that we have chemoreceptors or chemotaxis receptors that sense specific compound in root exudates, and that this sensing is required for colonization of the rhizosphere has been shown in many other PGPR. For example, it's been shown in Bacillus subtilis, Bacillus amyloliquefaciens, Pseudomonas putida, Azorhizobium cholinodens, Cynorhizobium meliloti. And as you can see, or perhaps if you know a little bit these organisms, these are genetically tractable, and they also can be manipulated in the lab. They can be labeled fluorescently. So these studies have been done using model system. However, my lab was interested in developing a system that could allow us to detect chemotaxis of any soil bacteria without having to genetically manipulate them. So to do this, we partner with Metro Laser to test and develop a digital holography microscopy method to track in real time and in a high throughput format any motile organisms. And so digital holography microscopy basically uses diffraction patterns of particle in a volume to detect this particle within that volume. And so what you can do is take hologram over time of cells in a volume with a gradient or without a gradient. And you can generate this diffraction pattern, you can detect this particle, and then if you link this 3D information of particle tracking over time, you actually can generate tracks for hundreds of cells at once. You can even then extract individual tracks and determine how mixture of cells or cells in a gradient would behave, and you can see different swimming pattern, etc. So this was simple way we establish this, but recently we've been doing a lot more with a prototype assay, trying to see this 3D trajectory. And we are now applying this to applying different plant microbe association, looking to root exudate. But what is also particularly interesting is how long you can track bacteria. And here I'm going to show you a short movie where you can see the reconstruction of the diffraction pattern. Here, this is a track of a bacterium that you're going to follow on this movie. And these are the quantitative analysis. And the reason I'm showing this is because we can produce a lot of data that would allow us to characterize the behavior and response of bacteria to any attractant or repellent or any environment. And it's easy to do because, again, as I said, in chemotaxis, if you know how often a cell change swimming direction or how fast they go, you actually can tell whether or not they like their environment. So here is a real-time movie where you can see the tracking going on over time, and we get also the measure of turn angle or the speed of the cell and the 3D reconstruction on the bottom right hand. So this is quite a promising method, and it's been employed up to date by biophysicists interested in the detail of molecular mechanism of or biophysical condition of flagellar motility. But here we aim to really try to bring it to the more general public and to try to make it usable to characterize a lot more plant microbe association and motility. So with that, I'd like to thank my lab and all of you for listening. And I hope I whet your interest about learning about chemotaxis, motility, and this amazing plant growth promoting bacteria. Thank you.